Hello friends, welcome back to our new video for MCQs on biotechnology, and this is the part 3 of the series of videos on MCQs on biotechnology. So the first question of this video is, which of the following complex binds to promoter region of E. coli and stimulate transcription of operon? So the options are option A, SAP CRP complex, or option B, CAP CAMP complex, or option C, CAMP SAP complex, or option D, NAD CAP complex. So the right answer is option B, that is CAP-CAMP complex. Okay, so the CAP-CAMP complex binds to promoter region of E. coli and stimulate transcription of operon. Okay, so let's move on to the next question, that is question number 42. So what does CCC, that means triple C DNA, stands for? And the options are option A, covalently closed circular DNA, option B, covalently cleaved DNA, option C, complementarily cleaved circular DNA, or option D, closed circular cleaved DNA. So the right answer about the CCC DNA stands for option A, that is covalently closed circular DNA is known as CCC DNA. Okay, so let's move on to next question, that is question number 43. So cDNA is actually a kind of, option A, that is complex DNA molecule, option B, corrupt DNA molecule, or option C, DNA synthesized from mRNA, or option D, compulsory DNA of the cell. So the right answer is option C, that is DNA synthesized from mRNA is known as cDNA. Okay, so next question देखेंगे. Next question is 44. So 44, question number 44, the phenomenon utilized to separate cells like cancerous versus normal cells is called, option A, centrifugation, option B, malpractices practices, or option C, cell cider, or option D, cell sorting. And the right answer is option D, that is cell sorting. So the phenomenon utilized to separate cells like cancerous cells versus normal cells is called cell sorting. Okay, so let's move on to the question number 45. So the binding of metal cation by atoms possessing unshared electrons is referred as, option A, salt bridges, option B, chelation, option C, Tyndall effect, option D, cation effect. And the right answer is option B, that is chelation. So the binding of metal cation by atoms possessing unshared electrons is referred as chelation. So next question क्या है? Next question है question number 46. So an organism consisting of tissues or parts of diverse genetic constitutions is or are referred as, option A, hybrids, option B, chimera, or option D, hybrids, or option C, hybrids, or option D, western organisms. So the right answer is option B, that is chimera. So chimera is the right answer. So an organism consisting of tissues or parts of diverse genetic constitutions is or are referred as chimera. Okay, so next question क्या है? Question number 47. So heterotic effect in a hybrid is due to, option A, over dominance at most loci, or option B, non-allelic interaction, or option C, inter-allelic interaction, or option D, additive gene effect. So the right one is option B, that is non-allelic interaction. So heterotic effect in a hybrid is due to non-allelic interaction. So next question क्या है? Next है question number 48. So which one of the following microbe provides a natural system for gene transfer to produce transgenic plants? And the options are Pseudomonas syringae, option B, E. coli, option C, Agrobacterium tumefaciens, option D, Bacillus thuringiensis. And the right one is option C, that is Agrobacterium tumefaciens. So which one, that is Agrobacterium tumefaciens, of the following microbe provide a natural system for gene transfer to produce transgenic plants? So for transgenic plants we use Agrobacterium tumefaciens. So next question is 49. So transgenic plants are produced by introducing new genes by using option A mutation technique, option B crossing over, option C radiation, option D gene gun, and the right one is option D, that is gene gun. So transgenic plants are produced by introducing new genes by using gene gun. Okay, so let's move on to the question number 50. So question number 50 is the method by which scientists introduce gene into DNA of new organism via gene repair mechanism is referred as option A genomics, option B chimeroplasty, or option C gene surgery, or option D gene statistics. And the right one is option B, that is chimeroplasty. So the method by which scientists introduce gene into DNA of new organism via gene repair mechanism is referred as chimeroplasty. Okay, so next question क्या है? Question number 51. So 51 is fused protein from different species produced from chimeric DNA template is called option A novel protein, option B chimeric protein, option C proteomics, option D hybrid protein, and the right one is option B, that is chimeric protein. So fused protein from different species produced from chimeric DNA template is called chimeric protein. Okay, so next question, question number 52. So enzyme used to make cheese from milk is referred as option A chitin, option B chymosin, option C crematase, option D diastase, and the right one is option B, that is chymosin. So enzyme used to make cheese from milk is referred as chymosin. Okay, so next question is the assay used to detect effect of relative configuration on expression of two gene mutations is called option A bioassay, option B Ames test, option C Cistrans test, or option D gene separation. And the right one is option C, that is Cistrans test. Cistrans test. Okay, so the assay used to detect effect of relative configuration on expression of two gene mutation is called Cistrans test. So let's move on to the next question, that is question number 54. So the region of gene of DNA that encodes the amino acid sequence of a protein is called floating gene, or option B coding sequence, option C amino terminal gene, option D active gene, and the right one is option B, that is coding sequence. Okay, so the region of gene of DNA that encodes the amino acid sequence of a protein is called coding sequence. Okay, and the another one is called non-coding sequence. So move on to the next question, that is question number 55. So the protein consisting of metal or inorganic prosthetic group or both is a kind of transgenic protein, that is option A, or option B conjugated protein, option C prosthetic protein, or option D incomplete protein, and the right one is option B, that is conjugated protein. So what is conjugated protein? So the protein consisting of metal or inorganic prosthetic group or both is a kind of conjugated protein. So let's move on to the question number 56. It is the nucleotide sequence which gives the most common nucleotide at each position where number of variations in nucleotide sequence occurs is nothing but option A mutant sequence, option B consensus sequence, option C conjugated sequence, or option D all of the above, and the right one is option B, that is consensus sequence. So the nucleotide sequence which gives the most common nucleotide at each position where number of variations in nucleotide sequence occurs is nothing but consensus sequence. Okay, so next question is question number 57. So the form of DNA that present at 75% relative humidity and requires presence of sodium, potassium, or calcium ions is option A Z form of DNA, option B aDNA, option C bDNA, option D tIDNA, and the right one is option B, that is aDNA. So the form of DNA that present at 75% relative humidity and requires presence of sodium, potassium or calcium ions is aDNA. Okay, so let's move on to the question number 58, that is the catalytic antibodies of synthetic construct are referred as option A catabodies, or option B novel enzyme, option C abzymes, option D idiotype of antibodies, and the right one is option C, that is abzyme. So what is abzyme? The catalytic antibodies of synthetic construct are referred as abzymes. Abzymes are which antibodies that has catalytic activity. Okay, so let's move on to the question number 59. So those enzymes within microorganisms that populate extreme involvement are option A abzymes, option B thermophilic enzymes, option C extremo enzymes, or option D detritus enzyme, and the right one is option C, that is extremo enzymes. So those enzymes within microorganisms that populate extreme environment are extremozymes. Okay, so the last question of this video is the mitochondrial enzymes that catalyze the incorporation of iron into the protoporphyria molecule are option A ferrochelatase, option B chelation, option C ferritin, or option D chelation. Okay, so it is a repeat question, repeat option. So the right answer is option A, that is ferrochelatase. So the mitochondrial enzyme that catalyze the incorporation of iron into the protoporphyria molecule are ferrochelatase enzyme. Okay, so these are the all 20 questions of this part 3 video on MCQs on biotechnology. Okay, so thank you for watching this video.